How's it going, everyone? So in today's lecture, we're going to start with the male reproductive system. Kind of a lot of information in this, and hopefully this video won't take too long. I'll try to go a little bit quicker. So first thing I want to talk about, which you may have already been familiar with, but is the idea of haploid versus diploid. Um, we know that as adults, as full-grown humans, we have two pairs of 23 chromosomes. We get 23 from mom, 23 from dad, so that in your body cells, you have a total of 46. Um, the male gamete is the sperm, and the female gamete is the egg. Each of your gametes has 1/2 the amount of chromosomes, so 23. Obviously 23 in the sperm from dad, 23 in the egg from mom. Now, the gametes are said to be haploid. Okay, so you can just remember it as having half the amount of DNA, and when you put them together, you have diploid, which is twice the amount of DNA. Humans overall are diploid organisms, and the gametes are haploid. So just keeping that in mind, it's a very basic topic, but something just to keep in mind when you hear the terms diploid and haploid, to remember your body cells are diploid, but your gametes are haploid. The function of the male reproductive system is to produce sperm and transfer them to the female reproductive tract, right? That's the whole point, is to reproduce, and in order to reproduce, we need a sperm and an egg to combine. The testes produce both sperm and androgens, which are hormones. In this case, we're talking about the sex hormones that support male reproductive physiology. In humans, the most important male androgen is testosterone. There are several accessory organs and ducts that aid the process of sperm maturation and transport the sperm and other seminal components to the penis, which then delivers sperm to the female reproductive tract through ejaculation. So let's talk about all these different parts. The first part is the scrotum. The testes are located in a skin-covered, highly pigmented, muscular sac, for lack of a better term, called the scrotum. And the scrotum extends away from the body and behind the penis. The location is important in sperm production because sperm production proceeds more efficiently when the testes are kept about 2 to 4 degrees Celsius cooler than core body temperature. The scrotum can also contract using two types of muscles, dartos and the cremaster muscles, and that can elevate the testes if they're cold, either in cold weather or being in water. That moves the testes closer to the body, increasing the heat and decreasing the surface area. And then alternatively, they can relax if the environmental temperature increases, and that will move the testes farther away from the body core, increasing surface area, which promotes heat loss. So it's very important to keep the testes cooler so that the sperm that are produced are efficient and healthy. Okay, so that's the scrotum, which holds the testes. What are the testes in general? The testes, or singular testis, are the male gonads, which are the organs that produce the male gametes, which is sperm, and the androgens, which I just said are hormones, and the important one is testosterone. They are surrounded by two distinct layers of protective connective tissue. The outer tunica vaginalis, and beneath that is the tunica albuginea, which is a tough, white, dense connective tissue layer covering the testes itself. Not only does the tunica albuginea cover the outside of the testes, but it also invaginates to form septa that divide the testes into 300 to 400 structures called lobules. Within the lobules, sperm develop in the structures that are called the seminiferous tubules. So just bolded it right there for you. Sperm cells develop in structures called seminiferous tubules. During the 7th month of the developmental period of the male fetus, each testis will move through the abdominal musculature and descend into the scrotal cavity. That is called the descent of the testes. And of course, there are a whole bunch of ways in which that doesn't work out correctly, and that is called cryptorchidism. It's the clinical term used when one or both of those testes fail to descend into the scrotum prior to birth. Okay, let's talk about those seminiferous tubules, because there are some 6 different cell types that we need to go through. Those include supporting cells called sustentacular cells, as well as 5 types of developing sperm cells called germ cells. Germ cell development progresses from the basement membrane at the perimeter of the tubule toward the lumen. So the first are the Sertoli cells, and the Sertoli cells surround all stages of the developing sperm cells. They are elongate and they are branching. They are a type of supporting cell called a sustentacular cell or a sustentocyte, and they're typically found in epithelial tissue. They secrete signaling molecules that promote sperm production and can control whether germ cells live or die. They extend physically around the germ cells from the peripheral basement membrane of the seminiferous tubules all the way to the lumen. They have tight junctions between the sustentacular cells, and that creates the blood-testis barrier, which keeps blood-borne substances from reaching the germ cells, and at the same time keeps the surface antigens that are on developing germ cells from getting into the bloodstream, which would promote an autoimmune response. Okay, then the germ cells. The germ cells, the least mature of them, are the spermatogonia, or if you talk about one, the spermatogonium, and they line the basement membrane inside the tubule. They are the stem cells of the testes, and that means that they're able to differentiate into any variety of different cell types through adulthood. So if I took the stem cell out and put it in a specific set of circumstances, it could change into a liver cell or a brain cell or a lung cell. That's what these stem cells are able to do. Spermatogonia divide to produce primary and secondary spermatocytes, then spermatids, and then finally spermatids produce formed sperm. The process that begins with spermatogonia and ends with production of sperm is called spermatogenesis. So spermatogenesis is just the genesis of sperm. Here's spermatogenesis. It occurs in the seminiferous tubules. We already said that, and those seminiferous tubules form the bulk of each of the testes, and it begins at puberty and it continues throughout a man's life. So if you remember, we talked about how men have a much longer window of reproductive viability than do women, because women, what we'll find out, have a finite number of their sex cells, whereas men continuously produce them throughout their life. The entire process, going from spermatogonia to sperm, actually lasts 64 days, but this is happening billions of times at any one moment, so men are never really at any loss of sperm cells. A new cycle will begin about every 16 days, but that's not consistent. You could have one happening right now and the next day and the next day and the next day, but for one individual seminiferous tubule, a cycle might begin every 16 days. Sperm counts are just how much sperm a male is making. It begins to decline a bit after age 35, but also, and probably more drastically, with the number of lifestyle choices. The big one is like smoking causes a huge decline in sperm count. And there's various other things as well. Here I just wanted to give you a look at a figure that talks about spermatogenesis, and I wanted to point out here, check out how the spermatogonium is labeled as 2N. By the way, this means diploid, and notice that we know that we have to get to sperm, which is a singular haploid. And so what happens is spermatogonium first go through mitosis. If you know anything about mitosis, mitosis starts with a diploid cell and ends with 2 identical diploid cells. Now, one of the spermatogonium becomes the primary spermatocyte and the other becomes the secondary spermatocyte. The secondary spermatocyte goes back and reverts to being a spermatogonium, whereas the primary spermatocyte then goes through a process called meiosis, and in meiosis there are 2 divisions, and what happens is you start with a diploid cell. 2N just means 2 copies, 1 copy. You start with a diploid cell, you do 2 divisions, where you end up with 4 haploid cells, okay? And so I just wanted to point that out, that spermatogenesis begins with diploid cells that go through mitosis, one returns to the beginning, and then one diploid cell then goes through meiosis, which is the process of going from diploid to haploid. Now, there are a number of ways in which we will see that meiosis could go wrong and end up with an incorrect number of chromosomes in the gamete. And then when that gamete fuses, the developing zygote will have the incorrect number of chromosomes, and that leads to a number of different issues, some that can be brought to term, some that cannot. But anyway, let's keep moving. Here is the structure of a formed sperm. By the way, just pointing out that a sperm is much, much smaller than almost every other body cell. I'll probably show you at some point a sperm, not in this lecture, but in the next lecture when we talk about female reproductive system, I'll put in a picture of what a sperm looks like compared to an egg cell, and it's just incredibly different. Sperm are really just made to do one thing, and that is to swim to a female egg cell. They're divided into the head, which contains the DNA, and basically just the DNA. So in here is the 23 chromosomes, just a little bit of cytoplasm and not a whole bunch else. Then there's this mid piece here that has a bunch of mitochondria, and we know what mitochondria do. Mitochondria produce energy for the cell to use, and that cell, those mitochondria use the stored fructose, so not glucose, but fructose, that in order to produce the energy that can whip this flagellum, which is the tail, in order for this cell to move, to provide motility. And so basically that's the structure of a formed sperm. You guys probably have seen a picture of sperm before, but just to go over it again. Let's talk about how that sperm gets transported. To fertilize an egg, sperm have to be moved from the seminiferous tubules, where they're made, through the epididymis, and later, during ejaculation, along the length of the penis and out into the female reproductive tract. The epididymis is a coiled tube that's attached to the testes, where newly formed sperm go and continue to mature. It's approximately 6 meters or 20 feet in length if it was straightened out, but in the testes it's highly, highly coiled. It takes an average of 12 days for sperm to move through the coils of the epididymis, and the shortest recorded transit time was 1 day. It's just a little fact there. But as the sperm moves along the epididymis, they're constantly maturing. And basically it says here that they are moved along predominantly by the contraction of smooth muscles that are lining the epididymal tubes. And they, like I said, they'll further mature, and eventually once they get to the end, will have acquired the ability to move under their own power. And then once ejaculation occurs and they are inside the female reproductive tract, they'll use that ability to move independently toward the unfertilized egg. The more mature sperm are then stored in the tail of the epididymis, the final section, until ejaculation occurs. What happens, what we'll see, is that tons of sperm are ejaculated, and it's basically a race to the egg, to whatever singular sperm cell reaches the egg cell. Okay, let's talk about the ductus deferens. During ejaculation, sperm exit the tail of the epididymis and are pushed by smooth muscle contraction to a structure called the ductus deferens, also called the vas deferens. Because the vas deferens, or ductus deferens, is physically accessible within the scrotum, you can surgically sterilize this by interrupting the sperm delivery. And that's just done by cutting and sealing a small section of the vas deferens. That procedure is called a vasectomy, and it's an effective form of male birth control. Now, that process is technically reversible. However, doctors always say this is something that should only be done when a male has 100% gone through the process of deciding they do not want to reproduce. So just a little side note here, sperm make up only 5% of the final volume of the ejaculate, or whatever, the semen, what you call the semen. The bulk of the semen is produced by 3 critical accessory glands: the seminal vesicles, the prostate, and the bulbourethral glands. So the seminal vesicles. As sperm pass through the ductus deferens at ejaculation, they mix with fluid from the seminal vesicle. The seminal vesicles are glands that contribute approximately 60% of the semen volume. Um, this fluid contains large amounts of fructose, which I said gets used by the sperm mitochondria that allows them to move through the female reproductive tract. And that fluid, which now contains both sperm, seminal vesicle secretions, then moves into the associated ejaculatory duct, which is a short structure formed from the ampulla of the ductus deferens and the duct of the seminal vesicle. The paired ejaculatory ducts transport the seminal fluid into the next structure, the prostate gland. And the prostate gland is a centrally located gland, and it sits anterior to the rectum at the base of the bladder, and it surrounds the prostatic urethra, which is the portion of your urethra that runs within the prostate. It's about the size of a walnut, and it's formed of both muscular and glandular tissues. And what the prostate gland does is it excretes an alkaline fluid to the passing seminal fluid, which is now called semen, and that's critical to first coagulate, or basically clump together, and then decoagulate the semen following ejaculation. When you coagulate or make it thicker, that helps the semen retain itself within the female reproductive tract and not just fall out. And then that provides time for sperm to utilize the fructose to get moving. And then when the semen regains its fluid state, sperm are then ready to use that ATP to be produced to swim and pass further into the female reproductive tract. According to the Centers for Disease Control and Prevention, the CDC, prostate cancer is the second most common cancer in men. There are many different types of prostate cancer. Some are actually slow moving and wouldn't be detectable. Others are unfortunately very fast moving and very malignant and very deadly. Okay, next are the bulbourethral glands. They produce the final addition to semen. It's made by two of the bulbourethral glands, are called the Cowper glands. They release a thick, salty fluid, and that lubricates the end of the urethra and the vagina and helps to clean urine residues from the penile urethra. The fluid from those accessory glands is released after sexual arousal and shortly before ejaculation or the release of the semen. Therefore, sometimes it's called pre-ejaculate. And it's important to note that it is actually possible for this fluid to pick up some sperm that was already present in the urethra, and so therefore it might be able to cause pregnancy. Okay, the next we'll talk about the penis. It's the male organ of copulation, which is, of course, sexual intercourse. It is flaccid for most actions that are non-sexual, urination as one example, and turgid or hardened with sexual arousal. When the penis is erect, the stiffness of that organ allows it to penetrate into the vagina, deposit semen into the female reproductive tract. The shaft of the penis surrounds the urethra, which is external for men, of course. It's composed of three column-like chambers of erectile tissue that span the length of the shaft. The two larger lateral chambers is called the corpus cavernosum, or the corpora cavernosa, and together those make up the bulk of the penis. And then the third smaller one is the corpus spongiosum, which could be seen on the raised edge of an erect penis. It's a smaller chamber, and it surrounds the spongy or penile urethra. The end of the penis is called the glans penis, and it has a high concentration of nerve endings, which results in very sensitive skin that influences an increased likelihood of ejaculation. The skin from the shaft can extend down over the glans and forms a collar called the prepuce, or the foreskin, and the foreskin also contains a dense concentration of nerve endings and helps to both lubricate and protect the sensitive skin of the glans. There is a surgical procedure called circumcision, which is often performed for religious or health reasons or social reasons, removes that skin typically within days of birth. Okay, let's quickly talk about testosterone. It's an androgen, of course, which is a steroid hormone produced by what are called Leydig cells or interstitial cells. It's responsible for the secondary male sex characteristics, which are, of course, increased facial hair, deepened voice, all of these things. And the continued presence of testosterone is necessary to keep the male reproductive system working properly, and Leydig cells produce approximately 6 to 7 milligrams of testosterone per day. How do these hormones get controlled? Well, we get a release of a hormone called gonadotropin releasing hormone, or GnRH, from the hypothalamus in your brain. That stimulates the endocrine release of hormones from the pituitary gland. what you get is a binding of GnRH to its receptors on the anterior pituitary gland, which stimulates the release of two more gonadotropins, luteinizing hormone, LH, and follicle stimulating hormone, FSH. Both of those hormones are critical for reproductive function in both men and women. In men, follicle stimulating hormone binds predominantly to the Sertoli cells within the seminiferous tubules, which of course promotes spermatogenesis. And follicle stimulating hormone also stimulates the Sertoli cells to produce hormones called inhibins, which functions to inhibit follicle stimulating hormone release from the pituitary, thus reducing testosterone secretion. This works on a negative feedback loop where the presence of testosterone is the signal to either produce more testosterone or to produce less testosterone. And so with that, that brings us to the end. Sorry for a longer video there, but that is the male reproductive system. I'll be back with the female reproductive system in the next one.